more about space. So, V1 neurons have receptive fields, and neurons physically close tend to have receptive fields that are close to each other, or even overlap. Which means that neurons that are close to each other tend to care about the same physical space relative to the position of the center of gaze. This is the basis of the retinotopic organization for neurons in V1, as we just explained in the last section. Which is just to say that there is a map-like organization in V1 that mirrors the retinotopic space. Turns out, the receptive field size increases as we go up the visual hierarchy. V1 neurons have smaller receptive fields than those in V2, which in turn are smaller than the receptive fields in V4, and so on. Why do neurons increase in receptive field size as we move up the visual hierarchy? One way to think about it is this. A V2 neuron may receive inputs from multiple V1 neurons. So if each of these V1 neurons cares about one specific location, and the V2 neuron cares about the inputs from each of the V1 neurons, effectively that V2 neuron cares about all of those locations combined, which means a larger receptive field. And a V4 neuron, in turn, receives inputs from multiple V2 neurons, so its receptive field is even bigger. Although the receptive fields get bigger further along the visual hierarchy, the retinotopic structure is preserved throughout the visual areas. That is, each of the regions, be it V1, V2, V3, V4, MT, etc., all have their own retinotopic maps, which is to say, within each region there is a spatial organization that maps to the retinal space. This retinotopic organization, like we said, is true of dorsal stream areas like MT as well, but things get a little more interesting further into the parietal cortex. We don't usually say that parietal areas are sensory areas. Sometimes we call them association areas. But there are neurons in the parietal cortex which also respond to visual stimuli. The difference, though, is that many of these neurons are not retinotopically organized. Rather, they are spatiotopically organized. That is, instead of caring about stimuli in specific retinal locations, they care about them in the actual spatial locations. That's really interesting. Imagine you are looking at 3 dots like this. When you stare at the left one, 2 dots will fall into your right visual field. Some neurons in the visual areas of your left hemisphere will fire to signal the presence of these 2 dots on the right. Now, if you move your gaze towards the dot on the right, there will no longer be any dots in your right visual field. So those neurons firing will return to baseline level, which means when you make eye movements, these neurons firing will change a lot. These ballistic eye movements are called saccades, and we make them a lot, as frequently as about 3 times per second, even though you might have never noticed. Given these frequent saccades, the representation in early visual cortex must be rather unstable, which is perhaps why having the spatiotopically organized neurons in the parietal cortex may be handy. When you move your gaze from one dot to another, the dots obviously don't move in actual space. They only change their location on your retina. Accordingly, if a neuron cares about a location on the left, it will continue to do so even when you move your eyes. This may explain why we see the world as relatively stable even though we make eye movements all of the time. The fact that there is retinotopic organization throughout the visual cortex is also handy for scientists studying the brain. Because each visual area has its own retinotopic map, the boundary between areas can be clearly defined. Let's say we use fMRI to study these areas. If we sweep through the retinal space with a stimulus, we can see corresponding bold signal changes in a spatially ordered fashion. That is, if we present a flashing visual figure on a computer screen and move it around space in a particular order, we can predict changes in bold signal within each visual area in a corresponding way, allowing us to map out where these areas are exactly. This is very useful because it means that for visual areas, we don't have to use standard atlases to roughly guess where the region is, which may differ a little bit from person to person. Instead, we know exactly whether we are in V1 or V2 if we have done this retinotopic mapping procedure for the person being tested.